Okay, good afternoon, ladies and gentlemen. Welcome to my talk. My talk is about an overview of Pleosporales. I'm from Beijing Forestry University in China. Okay, as we all know that Pleosporales belong to Docidiomycetes, and it is a large order within Docidiomycetes. Besides its common features of Docidiomycetes, Pleosporales has been defined by the following features to distinguish from the other orders of Docidiomycetes. They are perisessioid, cleistosessioid, hysterosessioid ascomata, trabeculate or cellulose pseudoparaphysis, and as well as the DNA sequences that, of course, have been used in helping for taxonomy as well. In 1955, Pleosporales was proposed by Luttrell, and from 1955 to 1978, Pleosporales was defined having perisessioid ascomata, and the pseudoparaphysis can be trabeculate and or cellulose. And 7 to 8 families have been included during this period. From 1979 to 1999, Pleosporales have been considered having perisessioid ascomata, rarely cleistosessioid or hysterosessioid ascomata, and trabeculate, and the pseudoparaphysis is cellulose. And during this time span, 14 to 18 families have been included. From 2000 to 2008, Pleosporales was considered having perisessioid ascomata or rarely cleistosessioid or hysterosessioid ascomata, and the pseudoparaphysis can be trabeculate or cellulose, and the limited DNA sequences can also be available for identification. And during this period, 13 to 23 families have been included. From 2009 to present, Pleosporales is considered having perisessioid ascomata, rarely cleistosessioid or hysterosessioid ascomata. And the pseudoparaphysis can be trabeculate or cellulose, and the pseudoparaphysis sometimes can be absent. A large number of DNA sequences can be available now for identification. During this time, 40 to 35 families have been included in this order. In particular, ecological information is considered having taxonomic and phylogenetic significance. Pleosporales so far is containing 35 families, 139 genera, and 10,000 species. This group of fungi can be saprops in freshwater or maritime environment. They can also be saprops in forest leaf litters or on animal drops. They can be hemibiotropic or biotrophic in plants or animals, and some species of them can also be pathogenic or human beings. Okay, this figure is showing us the phylogenetic significance of lifestyles or habitats of Pleosporales. The green dot here means the hemibiotropic or biotrophic members of Pleosporales, and the red dot means the saprobic members which lives on terrestrial environment, and the blue dots means the aquatic the members that saprops in aquatic environment, which can be freshwater or maritime. And the black dot means the coprophilous members, and all the other, So we can see that most of the plant pathogens of Pleosporales nested in the suborder Pleosporinae, And here, this is the trematospheric, which can cause some species of trematospheric can cause disease on human beings. And this is the Massariaceae, which is considered as a weak pathogen of Acer or Rosati. And the 11 families of Pleosporales continue members that can cause plant disease or human disease. And 10 families live in aquatic environment, which can be in freshwater or marine environment. The 3 families that the B means that the families that are living on bamboo or some other monocots. So 3 families are bambusicolars, and 2 families are coprophilias, which means they are living on animal drops. From this figure, we can see that the lifestyles or habitats have phylogenetic significance at a family or higher level classification. This figure is showing us the phylogenetic distribution of reported species. Okay, the blue bar here means the amount of species that's reported in each family, and yes. So we can see that the suborder of Pleosporines containing 44% of the Pleosporales, and the other order, Massariaceae, containing 17% of the Pleosporales, and another 21 families containing 39% of the members of Pleosporales. So we can see that the pathogenetic groups are getting better studied than all the other groups. This figure is showing us the geographic distribution of the reported species. Okay, 50% reported species are distributed in Europe, which means 50% species of Pleosporales reported in Europe, 24% in North America, 12% in Asia, 9% in South America, 4% in South Africa, and only 1% in Australian region. So we can see that the geographic distribution of the species is quite, of reported species are quite uneven, and a large area of the world is poorly studied. This, in recent years, several studies have been focused on the genetic types of Pleosporales. So far, 113 genetic types have been described and illustrated. From the genetic types we have studied, we can see that 74% type specimens, the genetic type specimens, are in good condition, and detailed information about morphology can be obtained. And 11% the situation is okay, means that the basic information are still available, but 6% are in really bad condition, which means almost no information can be available anymore. And 9% specimens cannot be located, so we can consider that they are lost. In total, 15% of the genetic types no information can be available anymore. How about the cultures or sequences of the genetic types of the type species? In total, 43% of the types of the genus, that means the type species of the genus, have cultures connected, but only 55% of them can be verified as correctly identified. In total, only 23% generic type species have correct sequences connected. This figure is showing us the introduction speed of new genera and families within Pleosporales. In 1909, the first genus of Pleosporales, which is Pleosporales, was introduced. In the following 90 years, six families and 40 genera have been described. In the following 100 years, another 8 families and 65 genera have been described. In the following 10 years, which means from 2000 to 2010, another 10 families and 22 genera have been described. And in the following 4 years, which means from 2011 to 2014, 11 more families and 10 more new genera have been described. And so we can see that the higher level classification is changing rapidly, especially the family level classification changing more rapidly. However, there are still some unresolved families. For example, the Melanoma. This dendrogram is the most comprehensive dendrogram published till today, but is containing only 4 genera and 10 species included. But currently, 16 genera and 500 species included in this family. Another example is Pleosporales. In this phylogram, only 7 genera and 16 species included in the phylogram. But currently, 20 genera and 700 species included in this family. Other examples are Pleosporales, Pleosporales, and so on. So for further study, we should explore more species, especially species from poorly studied or rich area or those unresolved clades to find more taxa. And we should also use more correct DNA sequences for phylogenetic analysis to get a more comprehensive backbone phylogram. And also, we should use a polyphasic taxonomic method, which means we should combine the morphology, ecology, molecular data, and some other parameters to make a modern taxonomic classification. Okay, thank you very much.